All right. Well, what a crowd we have. Thank you so much for coming. And I guess the folks from Louisville set the record in terms of driving the farthest. Thank you so much. And then I have a whole crowd here from Sudbury, I understand. So appreciate it so much. I'm glad for your interest in this topic. And I will tell you by way of introduction, you missed the introductory lesson. And there's not any time today to go over that. If we did this properly, we'd do a review of that lesson first. Not time for that, so you can get a copy of it and look at it. We're going to proceed on today. The introductory lesson was designed to show why this topic is reasonably discussed from a scientific perspective. Many scientists think if you introduce intelligent design into the discussion, you've just stepped outside of science. So a big purpose of last night was to show that that's not so. Scientists use intelligent design all the time as a part of what they do. So that's the underneath background portion. I'll not say any more about that. Let's get to the subject at hand. We're going to look today at two different sources of scientific information that I believe can be interpreted from a creationist perspective more reasonably than a evolutionary perspective. So the first lecture is on design at the microscopic level, my favorite subject, because I taught chemistry for many years. So it's close to my heart. I know all of you love chemistry. It's your favorite subject. That and mathematics. Am I right? If it isn't, I'm sorry for you. So we'll be dealing with those elements today in this first lecture, both the science and the mathematics. And I'll do my best to keep you awake. And I will tell you like I told the class last night, and we're going to treat this like a class. If you start falling asleep, then we're going to stop and everybody's going to stand up. We're going to do some calisthenics, and then we'll sit back down. I don't allow sleeping. Good to have you here. All right. Another thing I want to do, and I'll start that way because I forgot to introduce two books last night. Very good little book. I'm not going to deal with it specifically today. It's by Jerry Fodor and Massimo Piatelli Palmerini. Good Italian. The title of this book is What Darwin Got Wrong. And the theme of this book, What Darwin Got Wrong, is natural selection. And they're taking the position that natural selection does not explain what they claim it explains. They are evolutionists through and through, not creationists. And they're saying what Darwin got wrong was that he thought natural selection explained how everything evolved. And they're saying, no, it doesn't. Isn't that interesting? It's only the most important thing Darwin's credited with. And they're saying he got it wrong. Interesting study. Let's see. Here's another brand new book. Just came out this year. It's called Darwin's Doubts. Written by the same author as the book we're going to look at today. Darwin's Doubt. I'm about 2/3 of the way through. I was reading with a passion this morning, right? Trying to get through before 1:30, but I didn't make it. Fascinating book. It is about the Cambrian explosion in the fossil record and the implications that has for the evolutionary picture of life. It impacts everything. And Stephen Meyer, a graduate of Cambridge University, the same university Darwin attended, and several other universities, is a brilliant man in my view, and he is an intelligent design theorist, but he knows his stuff. I highly recommend that book to you as well. 2013 copyright date. The others we're going to use today, so we'll get to them in just a minute. I also will make a comment that I'm leaving a bibliography with William of all the books that I reference and a whole lot more that you're welcome to have as well. And one of the exciting things of the time in which we live is that there's more good material been published about this subject by well-respected and published scientists from a creationist standpoint than all the history of mankind put together. You're really blessed. So if you want to dig into this subject, you live in the time to do it. And I put 4 stars by the best books. So that's where you start. You're going to use my bibliography. All right. I'm going to refer to one biblical text as a beginning place. And I do that for this reason. Bible believers sometimes object to this approach, the approach I'm taking today. They think you should use Bible as you approach this subject. And I'm not opposed to that. But if you're talking with someone who doesn't even believe in God, it does no good to quote Scripture to them. You have to start back from that. So the approach I'm taking takes us back beyond a scriptural approach. I'm using this verse because I want Bible believers to know that the Bible itself uses the same approach that I'm taking. And Romans 1:20 is the classic passage in Scripture that refers to that. So it says, I'll just read it to you. Should have had my Bible open to that already. Romans 1 verse 20 says, "For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse." What does it say? It says God is invisible, but you can still see him. So how do you see him? You see him in the things that are made. And the things you should be able to see by examining the things that are made is his eternal power, must be a tremendously powerful being, and secondly, his divine nature, that is, he's not part of nature. He's separate from nature. Those things you should be able to conclude from examining the natural world. And it's the famous argument from design, which is exactly the argument I'm using in every one of these lectures. The argument from design. Can you see okay? I feel like I'm blocking your view. You're welcome to move. All right. So that's what I'm going to do in this lecture also. It's the argument from design. We're going to look at things that are made. They're going to argue for a maker, an intelligent designer. So we started last night with a website for the SETI organization. Search for Extraterrestrial Intelligence. So I think I'll have a raising of hands here tonight, this afternoon. How many of you in this audience believe there are extraterrestrial intelligences out there somewhere? Nobody. You think maybe? Possible. Hard to know, isn't it? These folks are looking for it. Signs of extraterrestrial intelligence. So what are they looking for? Here's what they say. SETI, the search for extraterrestrial intelligence, is an exploratory science that seeks evidence of life in the universe by looking for some signature of its technology. So they're looking into outer space, and they're looking for some signature of the technology of an intelligent civilization. So what are they looking for? They're looking for radio signals. Basically. And the kind of radio signals they're looking for are narrow-band radio signals that contain information. And one of the things they would tell you is if they received a signal from outer space with a language in it that we could decode and read, that would be excellent evidence that came from an intelligent source. I'd agree with that. And I think it's a perfectly legitimate scientific enterprise. Though I don't want my tax dollars paid for it. Because they haven't found one scintilla of evidence to this date. And they've been looking for about 50 years now. But they're still looking. You can go look on their website if you like. It's kind of an interesting exercise. There are radio telescopes all over this earth that are being used in a privately funded project to seek for signs of intelligence. Now listen to me, ladies and gentlemen. This is a very important point. Why can't we use that same kind of reasoning right here on earth? I think we can. And I would argue there are things that are shouting intelligent design to us right here on earth. And we can use the same kind of reasoning that they're using. So let's do that. What are some of the signs of intelligent design right here on earth? Well, I say every cell is full of signs. design. Cells look like things that are made, including a language, which we can decode and read. We've learned how to read cell language. All of you know that? Anthony Flew, I quoted last night, professor of philosophy, former debater, atheist, author. In his 80th year, he made this statement. It now seems to me that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design. And Dr. Flew, who formerly debated creationists and belittled the creationist view, has become a creationist. Now, he's not a Bible believer, but he's become convinced DNA and the things associated with it had to have a designer. And I appreciate such an honest man who in his 80th year changes his mind about something as fundamental as that. And he's taken a great deal of abuse from his fellow philosophers and the scientific community, I can assure you. Research on DNA continues to reveal signs of design everywhere. Anybody in this class know when the structure of DNA was first discovered and they got a Nobel Prize for it? Anybody know? Crick and Watson. That's it. Good. You know what year? Close. A little farther, a little later than that. I mean, there was research going on in 48, you can be assured. It was 1953 that they announced officially. And by the way, can I get off my subject here just a little bit? We say Francis and Crick. Dr. Francis Crick and Dr. James Watson. And we say Crick and Watson are the ones that discovered the structure of DNA. May I disabuse you of that idea? Crick and Watson just did the last little piece of it. They were building on the shoulders of giants. Lots of other folks have done a great deal of research, untold hours and months and years of study. Crick and Watson just happen to be the young bucks who kind of put us over the top. And they get all the credit. Not right. It's the way it is. But 1953 was a seminal year, folks. We're in 2013. That's 60 years later. In the last 60 years, we have learned more about how life functions at the molecular level than in all the history of mankind put together prior to that. You live in an exciting time. I want to share some of it with you. Anthony Flew said, I have to go where the evidence leads. I want to show you some of the evidence. And then you're going to have to answer the question, which is more reasonable? Does this look like it was designed by a designer? Or do you think nature did this by the powerful forces of physics acting upon matter? That's the question, because those are the only two answers. So where do you come from? There's only two possibilities, ladies and gentlemen. You either came about by natural causes. Life in all its forms emerged from simple substances by natural means. That's the evolutionary view. Or you came about by design. Life in all its forms is the result of intelligent shaping of matter. Let me put it in an example for you. The Sistine Chapel over in Rome is a magnificent painting on the ceiling of that chapel. You think that came about by natural causes? You know, you just take a bunch of paint, plaster, and you produce a Sistine Chapel. No, nobody believes that. We believe it took an intelligent designer by the name of to paint that. So he had the materials, but it took an intelligent designer to put them together in such a way to produce that. Same thing is true, I'm convinced, of living things. Life in all its forms would never have taken place, I believe, if there hadn't been an intelligent designer acting upon it. But those are the only two answers, folks. Now we're going to look at life as we know it, at the cellular, molecular level. Are you ready for a chemistry lesson? Good. I hope you got a good nap before you came, because I need your full attention. This is the book I'm going to be referring to considerably. Of course, this is just a summary of what we've learned. Stephen Meyer, the same one that wrote Darwin's Doubt. This came out in 2009. Pretty current, up to date, pretty thick. It's full of fascinating information, including the history of this subject. Stephen Meyer is a wonderful writer, even though he's also a very well read and studied scientist. A lot of scientists aren't good writers, but he is. And the question is going to be, when we look at all this evidence that we're going to present to you today, which is the more reasonable explanation? Natural causes or intelligent design? Those are the only two answers. And the question will be, which is more reasonable when we examine this evidence? All right. So, class, let's talk about chemistry. Life as we know it. And I'm going to refer now to a little book called Life Itself, Its Origin and Nature by Dr. Francis Crick, co-discoverer of the structure of DNA. I would say Dr. Crick knows about as much as anybody about life. So, what I'm going to tell you here in the next few minutes is how he described life in his little book. I'm going to get my glasses out so I can read it. All right, so I'm on page 72 of this little book, and here's what he says. Life as we know it on Earth appears as, by the way, can I interrupt that quote just a minute? When he says life as we know it on Earth, may I say to you, ladies and gentlemen, that's the only kind of life we know anything about? Life as we know it. There isn't any other kind of life that anybody knows anything about. There are books written about what other forms of life might be like, but nobody's ever seen them. Life as we know it on Earth appears as a synthesis of two macromolecular systems. The proteins, because of their versatility and chemical reactivity, do all the work, but are unable to reproduce themselves in any simple way. So one of the great class of molecules is protein. Structural materials are proteins. They make up the shape and mobility of living things. So we call them the structural materials. They're proteins. The tools and machinery are also proteins. They're called enzymes. But an enzyme, ladies and gentlemen, is a special kind of a protein. And that particular special kind of a protein's job is to speed up reactions. You have thousands of them in every cell in your body. And if you didn't, you would be dead. Because all the reactions would be taking place so slowly, you couldn't survive. But enzymes, ladies and gentlemen, and let me get you to repeat this, enzymes are what? Say it out loud. Proteins. Protein. Extremely important in every living thing. In fact, it's one of the macromolecular systems. Protein. And they maintain the metabolism, that is the growth and maintenance of living things. You with me so far? Now let's go on in the quote here. The nucleic acids seem tailor-made for reproduction, but can achieve rather little else compared to the more elaborate and better equipped protein. So the second great class of molecules, ladies and gentlemen, is the genes, the nucleic acids. They're called RNA and DNA. Ribonucleic acid and deoxyribonucleic acid. You got that? They are primarily fit for reproduction. So they contain the information and the directive function as a class of molecules in living things. Now, to help you understand that, well, let's just review here. There's two basic kinds of proteins. They're structural and they're enzyme. Structural and enzyme. I already said that, didn't I? Good. And they are the workhorses of living things. They do the work. All right. I'm going to spend probably 5 minutes on protein. The proteins deserve 2 years. And we're going to spend 5 or 10 minutes. So I apologize to every protein for the inadequate, incomplete description I'm about to give. When I get finished, I will not have touched the veritable hem of the garment of what we know about protein. So please understand, this is it's a light treatment. Here you have before you the 20 common amino acids that occur in living things. I'm going to give you a minute to look at those. Because at the end, there will be a quiz. If you were in my chemistry class, you would memorize every one of them. That's how important they are. Not only would you memorize their names, but you'd also memorize their chemical structures. Because the chemical structure is what gives them their properties, which allows them to do what they do in living systems. Now, why are they called amino acids? Let me show you. Every one of them has an NH3 group. That's called amine. So they're amino. If you'll notice, every one of those structures has an NH3 group. You see that? Every one of them also has a COO group. That's an organic acid. COOH is what it really is. So they're amino acids with an amine group and an organic acid group. Every one of them. And then they have other things attached to it. Those 20, ladies and gentlemen, are key to every living thing. Every living thing. Because the fact is that all living things that have proteins in them, which they all do, the proteins are built out of those. These are the building blocks for making protein. That's why you should memorize it. So let's name them. I'm not going to name all of them. Let's go through a few. There's glycine, alanine, valine, leucine, isoleucine, proline, serine, threonine, aspartic acid, and so on. There's 20 of them. Have I paused long enough? Every protein is a combination of those 20. It's just exactly like the 26 letters of our alphabet. Every word in English is made out of those 26 letters. Right? It's some combination of those 26. There are no other letters in English. In protein language, the amino acids are the letters. And so you get strings of amino acids, and when you get finished, a string of amino acids is a what class? Protein. Now, this next quote comes directly out of Dr. Crick's book. I'm saying that, are you listening to me, class? I didn't say this. I don't want you getting me in trouble. He said it. I want you to listen to what he says. Whoops, how did that happen? That's going backwards. Sorry, folks. Here's where we are. DNA and RNA are the dumb blondes of the biomolecular world. Fit mainly for reproduction, with a little help from the proteins, but of little use for much else really demanding work. Sorry. For the blondes and the body. I'm not making any blonde jokes. That is what he said. And I like to put it up here because that's the one thing you're going to remember from what I said. The RNA and the DNA are the dumb blondes of the molecular world. They're good for reproduction and not much else. The fact is, listen to what he said. The problem of the origin of life, how did life get started? How did we get the simplest forms of life? The problem of the origin of life would be a great deal easier to approach if there were only one family of macromolecules capable of doing both jobs. Reproduction and the work that the protein. But life as we know it always employs two families. You have to have the proteins and you have to have the nucleic acid. You have to have them both. And neither one can function without the other. That's a problem for an evolutionist, as Dr. Crick admits. This may well be due to the fact that no macromolecule exists which would conveniently carry out both functions. And so he proceeds to talk about how maybe this did evolve. Now, lest I forget later, I have to tell you that in this little book, he talks about maybe how that did evolve, but he realizes there's a problem. And maybe there wasn't enough time on Earth to evolve the first simple living forms in 4.5 billion years. So rather than turning to the possibility that there was an intelligent designer, what does Dr. Crick do? He spends the rest of this book talking about directed panspermia. And you don't have a clue what that means. It's a fancy name for saying somebody sent it here on a spaceship. He talks about what the spaceship might have been like. This is the Nobel Prize winner, people. Is there one scintilla of evidence that a spaceship sent the first simple life forms on Earth? Not one scintilla. But he spends the rest of this book talking about that possibility. Not God. We'd rather talk about some alien sending a spaceship here with some simple life forms on it than we'd want to talk about God. And that's our Nobel Prize winner, chemistry. It's incredible. Anything but God, whether there's any evidence for it or not, because it's a natural explanation, not a supernatural. That's Dr. Crick. All right, let's talk a little bit about the nucleic acid. We should have spent a lot of time on proteins. I want to spend my time today on DNA and RNA. Whoops, I did it again. Well, you get to see this again anyway. So, there's the famous structure that Watson and Crick discovered in 1950. It has a certain characteristic structure. It's a double helix with crossbars. Can you see that there's a strand wrapped around, and then here's a separate strand wrapped around, and they're twining around each other? And then they also have crossbars across here like this to connect those two strands together. Because of that structure, the DNA has the capacity to reproduce itself. Like the dumb blonde. Sorry. And my job for the next few minutes is to explain to you the chemistry behind DNA. I hope you've already heard most of this. Because this was well known in the 50s and 60s when I was in high school and in college. So what I'm talking to you right now is dated information. Here is a DNA molecule unzipped. See that? Here it is wrapped around each other. Here it is unzipped. You see this little blob right here? That's called RNA polymerase. Let me teach you, class. If a word ends in ASE in organic chemistry and biochemistry, that is an enzyme. Now, all of you were listening a while ago. An enzyme is a... Thank you. Say it louder. An enzyme is a... Yes. One of those important molecules that does all the work. That glob of protein sits right on the DNA and rides along it and allows it to unzip. Pretty amazing. And when it unzips, it exposes a side, in fact, exposes two sides. And that side that's exposed has a code on it, ladies and gentlemen. And we've learned what that code is. It's a language that we've learned to read. We know exactly what it means. And I'm going to tell you what it means in just a minute. But first I want to tell you how this works. You see, when it unzips, a copy is made. Do you see this blue line right here? That's an RNA transcript. It's called messenger RNA. It makes a copy of what's on the DNA. It's the exact mirror image. Do you see that the A there, let's see. Can't see that very well. But the C attaches to the G and the G to the C. The T to the A, the T to the A to the U. That U there on the RNA replaces the T on the DNA. Don't ask me why. That's a long subject. The U replaces the T. So every time you see an A on the DNA, you're going to see a U on the RNA. Every time you see a T on the DNA, you're going to see an A on the RNA. A and T go together, C and G go together, like love and marriage and a horse and carriage. You know why those things fit together like that? Because of their chemistry. They fit together. All right. Now what's important is that this series of bases, and I don't know if you can tell it, but there's exactly 4 of them. A, T, C, and G. Who in this class knows what that stands for? Anybody? Okay, I'm going to tell you. That'll also be on the test. A is adenine. C is cytosine. G is guanine. And T is thymine. Got it? Those are 4 bases that ride along the strand of a DNA. And notice, let's just read a little bit here, shall we? Let's read together. I'll say it and you just listen. ACG, TAG, GTA, CGT, TAC, GGC. Did you notice I was reading that in groups of 3? And the reason I was doing that is because we have learned that in the genetic language, every word is exactly 3 letters. Wouldn't that be nice? If every word in English just had 3 letters. It'd be a really simple language. In genetic code, every word has 3 letters. And we know exactly what they mean. So watch. As you come along this strand of DNA here, you can actually read a code off of here. The RNA makes a copy of it that can then be taken somewhere else and stuff is done with it. Okay. I pause every now and then to give your mind a chance to catch up with me. Because sometimes I talk at 150 miles an hour. All right, what I have just described to you, we should spend the next 2 weeks on. We will proceed. What you see before you is the genetic code. Let's see if I can explain it to you. Remember those 3-letter words I mentioned to you? Those 3-letter words are made up of those 4 bases, A, C, G, and T. Notice over here on this side, this is the first base, and U, notice, replaces the T. Across the top is the second base, and down this side is the third base. So with that combination, you can write every 3-letter word that's possible in the genetic language. Well, let's do it. We're going to practice a little bit here. If the first letter is U, and the second letter is U, and the third letter is U, that's this one right here. U means phenylalanine. So let me see if I can pronounce that word. Ooh means phenylalanine. You don't pronounce words in the genetic code. But if you did, it would be ooh. Let's try another one. How about C-U-U? C-U-U. That's this one right here. C-U-U means leucine. Who means leucine. Let's try another one. How about A-U-G? That's this one right here. That means methionine. It also means start. Does anybody get the pattern up here? What is phenylalanine, class? What is leucine? What is arginine? What is threonine? What is serine? What is that? That's the 20 amino acids you were supposed to memorize a while ago. Those three-letter words, ladies and gentlemen, are the codes for all 20 amino acids. In fact, do you see some repeats up here? Yeah, because let's do a little math. If you've got 4 letters to use, and you're making 3-letter words, so you're talking combinations here. How many words can you make if you've got 4 letters to use, and you can repeat, and you can make 3-letter words? So 3-letter words taken from 4 letters. How many can you make? Let me tell you how to do it, class. In the first position of your 3-letter word, how many possibilities are there? 4. In the second position, how many possibilities? No, you can repeat. So you can use 4. How about in the third position? 4. So how many words can you make? 4 times 4 times 4. Do that in your head. Thank you very much. That was fast. Good. We'll send you to the board. There are exactly 64 words up here, class. That's how many you can make using 4 letters, 3 at a time, with repeat. And every one of them codes for an amino acid. You have repeats. Now someone might say, well, why do you have to have repeats? I call it a backup system. It looks like a plan to me. All 20 amino acids are coded here, so that built into the DNA is a code for building proteins out of 20 amino acids. It's like a plan to me. In addition to that, as I already mentioned, there is a code that says start reading. That's AUG. You come across AUG, and it means start reading, and then keep reading. What else do you need, class? Don't you need something to tell you to stop reading? Did you notice there are terminators up here? Well, it actually says stop. See it? See, there are 3 codes, this one, and this one, that say stop reading. So built in this code, there's a code that says start reading and go get me a methionine. Read. Then you get to UAA and it says stop reading. And in between those two is a string of amino acids which makes a protein. Now let me tell you, class, I'm going to be asking you a lot of questions here in the next few minutes, and I'm going to tell you ahead of time the answer. It's protein. So don't miss any more questions. All right. So what is this a code for? It's a code for building protein. Strings of amino acids make proteins. Amazing. And I will tell you, if the SETI organization received a code from outer space, maybe I need to get out here. It's not wanting to go for me. Now let me say what I said again, so I lost your attention. If we received a message from outer space that just contained the genetic code in it, and we were able to decipher it and read it, what do you think everybody would say? There's an intelligent source from which we got that. There would be nobody that said, isn't it interesting what those stars did up there, blinking out the genetic code? Nobody would say that. Every newspaper on earth would announce we've received a message from outer space that's from an intelligent source. But the fact that we have it in every living cell, evolutionists say, isn't it interesting what nature did? Folks, there is zero evidence that nature can produce a language by itself that codes for specific things. But that's what we've got. Okay, let's see if we can make this work. Not working. You're going to try something else? Okay. In the meantime, we'll just do it the old-fashioned way. I will tell you, if we got the genetic code and a signal from outer space, every organization on Earth would say that was from an intelligent source. It's amazing to me that we don't see that when it's right here under our noses. Every living cell has that in it. But here's what I'll tell you, class. If I stopped right here, having shown you that every living cell has a language in it that we know how to decode and read, that should be enough to convince you somebody made that. That didn't come about by natural causes. Somebody made that. But that is just the veritable tip of the iceberg. Because life as we know it doesn't just have a language in it. It uses that language to do magnificent things. And my job now for the next few minutes is to explain to you how that code is used. And again, most of this was known in the 60s. All right, so here you have a little picture taken out of a biology textbook of what goes on in the building of proteins. You see up there in the top, let's see if I can at least make the light work. Yeah. You see up here's the DNA. Notice this is the nucleus of the cell, and see it has a membrane around it. Here is the DNA. That's that big long molecule I showed you about earlier. You'll notice that parts of it are being copied. Coded in the DNA up here is the code for messenger RNA, for transfer RNA, and for ribosomal RNA. I don't have time to talk about those other two. You think we should discuss them? Yes, it should take us another couple of months. We're not even going to have time to talk about that. I'm going to focus in on messenger RNA right here. Now, I want you to listen to me closely because I'm coming back to some of this later, and if you miss this now, you're not going to get what I'm talking about later. So listen. A copy is made of what's on the DNA, and it's released, and that's called messenger RNA. It makes a copy of the information up here. Then, do you see these little white areas in here? They are called introns. Those little white things. They have to be snipped out, and the messenger RNA made in its mature form before it migrates. So you copy it, you snip out the introns, you mature the messenger RNA, and then it migrates. Transcription, migration, translation. See if I got a little bigger picture here. That way. Here's a blow up of the transcription piece. Think this might work? All right. Does this have a light on it too, the middle thing? Is that a light? The middle? Okay. All right, see, that's a little bigger picture of it. Here's the DNA. It's being copied. And by the way, class, what is it that rides along that DNA that allows it to unzip? It is a protein. Thank you very much. That was very weak. What is it, class, that rides along the DNA and allows it to unzip? Protein. Thank you. Exactly right. So there's some protein involved right here. When you come down here and you have to snip out those introns, guess what it takes? Protein. Exactly right. They're called spliceosomes and editosomes. They splice it out and edit it back together. Proteins. Then you have your mature messenger RNA. You see, the DNA can reproduce itself, but it needs a little help. No, it needs a lot of help from proteins. Good. Okay, so now the messenger RNA is ready to migrate outside the nucleus. You see that little hole right there? The messenger RNA is going to go outside. Let's see, what do I hit? This top up here? These little top things? Oh, on the right and left. Okay. I'm jinxing this thing. I don't have time to wait. Do you see the messenger RNA coming out that hole right there? Here it is. it comes along down here into the cytoplasm of the cell, and it's picked up by two ducks hugging each other. Isn't that what that looks like? Those are called ribosomes, and they don't look like that. That's just a picture. But notice that the two ducts close in on this messenger RNA. You see the code on it here. And actually what's happening is that code is being read in the ribosomes, and out the bottom comes the protein. Amazing. Utterly amazing. So the code is transported to the ribosome, where that code is used to build protein. Looks like a plan to me. Whoops. All right. Right here's where we're going to take a break. You ready? I've got my helper here. I hope this works better than this does. All right. What we have here, ladies and gentlemen, is a little video clip from the website for this book. You can go home and get on the website. It's called signatureInTheCell.com. And you can watch this video clip anytime you want. It's sitting right there. But it's a neat little video clip showing you what happens in the building of a protein. And it's 3D, and I'm going to be the moderator. You got the sound turned off? I don't want to say what he said. I want to say what I said. All right. So, here's my guy running the machine. Please start it off. Do you see that we now have the DNA there? That's stopped. And you see the code on it, those 4 bases? And it's wrapped around like that DNA structure is. That's how this works. All right, now let's watch what happens. I'm going to go over here. Okay. So, here's the DNA strand. Now, as we watch it here, whoops, stop right there. You see this glob sitting right here? Class, that is a protein. Thank you very much. It's RNA polymerase. Remember, if it ends in ASE, it's an enzyme, which is a protein. Exactly. In fact, do you see the different colors there? It's several proteins bunched together. And what that thing does is it gets on the DNA strand and it rides along it. And as it does, it unzips the DNA and exposes the side. DNA can't unzip without a protein. Without several proteins. And by the way, let me get off subject here again. Have you ever had trouble zipping something back up? I certainly have. Do you know that can happen at the molecular level? It's sometimes hard to get DNA zipped back together. Did you know there's a specialized protein to help DNA zip back together? There is. Incredible. Sounds like a plan to me. All right, so go ahead now. Let's let this ride along here. You see, there comes another piece hooking up with it, and you got this nice, wonderful glob right along. Stop. That's more what it looks like. It doesn't look like that other thing. There's your glob riding along the DNA, and what's happening is, see this piece right here? That's your RNA that's being formed. Now, the next thing we're going to do is we're going to go down inside this blob, and we're going to look at what's happening down inside. So let's go. See what happens. Okay, stop. All right, here's the exposed side of the DNA. And so here are the bases. Maybe that's GAC, TAG, GGC, and so on. See that? Now what happens then is the RNA is actually formed piece by piece as it comes along and matches up with those codes. So let's watch it happen here for a little bit. See what's happening? It's coming up along the code and it's forming. Stop right there. Do you see these little pieces coming along here? They exactly match up with those codes and you form the messenger RNA. Guess what it takes to make that happen, class? Proteins! And by the way, there's one problem with this little movement. It is way too slow. But let's go ahead. It happens way faster than that. All right, so here you come along, and I guess you can stop. Stop right there. I meant for him to stop a minute ago. How does it know when to stop reading? There's a code that says stop. Otherwise, it'd just keep right on copying. Forever. There's a stop code. How does it know when to start reading? There's a start code. Incredible. Now, when you finish copying, it releases the RNA and it goes outside. You see it going out there? Okay, let's go. Here it is. There's your strand of RNA with exactly the right number of amino acids coded for in the first one to the last one. Oh, they're all right there. All right, then it starts migrating. Watch now what happens. And it migrates toward, you see those holes that's in the membrane of the nucleus. Here's the RNA migrating outside, and it comes to a hole. Now, folks, do you think any respectable nuclear membrane wants just anything passing through the holes? No, it does not. Only certain things are allowed through those holes. So you watch. As it comes up to the hole, watch what happens here. You see this hole right here? Here it comes. You see that little thing right there? Stop. That little circular thing? Guess what that is? Protein! It's the guardian of the gate. And it opens or closes depending on what's coming. Guess what it lets through? The RNA. And nothing else. So when RNA approaches, watch what happens. It nicely opens the door and allows the RNA to go through. Stop. And here it is coming out the other side. You see these holes on the other side? What do you think's guarding the gate on the other side? Proteins! Right. But do you think any respectable nuclear plasma wants something going in that way? Not without permission. Right. All right, now let's go ahead. So here comes our RNA strand outside the cell nucleus, wall there, and here it comes, and it is now being attacked by two ducks hugging each other. Except this is more what it looks like. And of course, whoops, I forget this. That's okay. We'll stop right there. Now we're down inside. And here's the RNA strand inside that little glob that you saw, and it's going across and it's being read like a tape through a tape player. And notice, here's the first three letters, CAC, AGA, CUA, CCG. Remember, class, that that's the exact mirror image message that it had up there in the DNA? So then when you bring up the matching one, this is exactly what's in the DNA. Because it's the mirror image of that. See? And this thing here is called transfer RNA. Did I mention that a minute ago? That's also coded up in the DNA. And did you know, class, that every one of the 20 amino acids has its own specific transfer RNA, which picks up that particular code and brings it in to the ribosome? Guess what it takes to make transfer RNA bring the reflected code into the ribosome? A protein. There's exactly 20 specific proteins for every one of those transfer RNAs. Are you keeping count of how many times you said protein? I should have told you at the beginning. Start getting your shoes off. Well, there's 20 right here. Now let's go ahead. Watch what happens now. You see? Stop. Now we have three-letter words that have been coded. The transfer RNA has come up, and look what's happening down here. These are the amino acids that have been brought in from your cell plasma. You better be eating your amino acids or you can't build proteins. Why do you think all the health nuts tell you to take your amino acids? Some amino acids are called essential because your body doesn't produce them. Others are called non-essential because your body produces them, but you better believe you got to have them. And they're floating around in your cell. Transfer RNA grabs one, brings it in here, exactly matching the code. And then you start building the Thank you. Because what are proteins? They're strings of amino acids. So there's your first amino acid, there's the second amino acid, there's the third amino acid. Now let's keep going. Watch what happens now. There's the third coat, it builds another one. Here it puts on another one. Stop right there. If you were in my chemistry class, you would learn exactly what kind of chemical reactions are taking place as those amino acids hook up. Those are called polypeptide bonds. And they're very specific chemical reactions that allow those amino acids to hook up to each other and make a string. Great. Now there's a problem with it. It is way too slow. So watch, they're going to speed this thing up. Try to give you some idea of how fast this happens. That's way too slow. Way too slow! Faster! Stop. Folks, I want you to watch me here just a minute. When you're building protein, just like I showed you here, how fast does it happen? You watch. That was about 10,000 times too slow. Yeah, that's good. I got his attention. Listen, people, the average enzyme can catalyze the reaction of about 1,000 proteins per second. You see that process that was happening in there as it hooks up one right after the other? Do you know how big an average protein is? How many amino acids in a row does this class think is an average protein that you have to all do? How many do you think? That's a little over. But there are ones that have 1,000,000. The average is 1,000. An average protein is 1,000 amino acids in a row. And these things can build that at a rate of thousands per second. Henry Ford in his Ford plant never touched this. Never touched it. And people say, isn't it interesting what nature did? The blind forces of physics produced this. If you can believe that, I got a bridge to say down in the Everglades. I can't believe it. And folks, if I may pause here again, when I get finished saying everything I'm saying today, we would not have touched the hem of the garment for what's happening in that cell. This is nothing compared to what's happening. All right, so here we are. We're now outside that ribosome complex. Do you see right here, there's your RNA that's being read. Do you see right there that little blob coming out? That's your protein being built. So let's watch. The RNA is going through there and being read. Here's the protein being built coming out the bottom. And stop. It's released. There's your protein. What you need to know, class, is that every protein has 3 realms of design. And I don't mind saying that. The first realm is called its primary design. That's the string of amino acids. And by the way, class, for every protein, that string has to be very specifically a certain order of amino acids. Just like, how about this in English? Anti-disestablishmentarianism. That's a specific word that has a specific sequence of letters. What about that's gibberish. It means nothing in English. It doesn't communicate anything. The same thing's true in genetic code letters. You have to have a specific sequence for each protein that is very specific, and if you don't get it right, it doesn't work. Class, that's the first level. That's called primary. That's hard enough. Every protein also has a secondary structure that's based on this primary structure because it's got pieces that stick out in different ways that give it a secondary structure. If you don't have that, you don't have a working protein. In addition to that, every protein has a third characteristic called its tertiary structure. Tertiary means third. In which it takes on a particular three-dimensional shape. And if it doesn't have that, it can't function. So watch. As it's been released here now into the cytoplasm, it is picked up by another organism. And inside that organism, it is shaped into its particular three-dimensional configuration that allows it to function as that specific enzyme or protein of other kind. See how it's rotating around there and forming into its three-dimensional shape? Stop. Guess what it takes to make that happen, Brian? Say it. Proteins! Are you getting tired of this? I told you the RNA and the DNA are the dumb blondes in the molecular world. Who does all the work? The proteins do all the work. So now once it's formed into its appropriate three-dimensional shape, it is released into the cytoplasm to go to do the work for which it was designed. And I love it when biology textbooks talk about the work for which it was designed, and then they say there's no design. Incredible. They talk about architecture. That implies an architect. All right. You did a good job. Can you huggle us back to the other? Can you believe this? With 2 flips of his fingers, you're back to the nose. Pretty amazing. All right. So we showed you then what happens. Pretty amazing. This is the progress toward producing protein. Isn't that overwhelming? That doesn't testify to a design to you. I don't know what I can do to help you. It's utterly amazing. For people to sit around and talk about how nature did that with the blind forces of physics acting on by natural selection is beyond me. Oh, it worked. I can't believe it. That was working too good, too well. All right, come on. All right. Here's the summary of the puzzle. I don't know if you caught this or not. DNA contains the code, right, class? It takes many proteins to transcribe and translate the code and build proteins. Would you agree? How many times did you say protein? It takes a bunch of them. These proteins are themselves coded in the DNA. Did you think about that? So let's talk about it. The very first one I mentioned that rides along the DNA is RNA polymerase, right? And makes it unzip. Without the RNA polymerase, you can't unzip DNA. Well, where'd you get that first RNA polymerase? It's coded in the DNA. How did you make the first one? Are you with me? You have to have RNA polymerase to unzip DNA. But RNA polymerase is coded in the DNA. This works a lot better than this. To produce these proteins from the code requires the proteins themselves. How could you produce the first RNA polymerase without an RNA polymerase already there? It's a problem. Which came first? The protein or the nucleic acids? It doesn't matter. You have to have them both. That's why Crick says in this book, the problem of the origin of life is because there's 2 large groups of molecules that you have to have both. There are over 50 proteins, ladies and gentlemen, used to make one protein. And all 50 of those are coded in the DNA. And all of them had to be produced by the same mechanism. So, Karl Popper says in Studies on the Philosophy of Biology, what makes the origin of life in the genetic code a disturbing riddle is this: the genetic code is without any biological function unless it is translated. That is, unless it leads to the synthesis of proteins whose structure is laid down by the code. But as Monod points out, the machinery by which the cell translated the code consists of at least 50 macromolecular components, otherwise known class as proteins, which are themselves coded in the DNA. Thus, the code cannot be translated except by using certain products of its translation. This constitutes a really baffling circle, a vicious circle, it seems, for any attempt to form a model or theory of the genesis of the genetic code. I say, amen. How did it get started, class? It is the old chicken and the egg problem at the molecular level. You have to have it all. Looks like a design to me. A living cell. Notice who said this, class. Sagan. This is an old quote from 1974. Now in 2013, we can increase this dramatically. But I can't get a Sagan quote anymore, so he's dead. So I like this one. A living cell is a marvel of detailed and complex architecture. You see that? That's because I believe there's an architect. He does it. Seen through a microscope, there's an appearance of almost frenetic activity. Now there's a good word. Frenetic. Who knows what that means? Exactly. I'll illustrate frenetic. That's frenetic. You look down inside a living cell, that's what it looks like. On a deeper level, it is known that molecules are being synthesized at an enormous rate. Almost any enzyme which classes a protein catalyzes the synthesis of more than 100 other molecules per second. In 10 minutes, a sizable fraction of the total mass of a metabolizing bacterial cell has been synthesized. The information content of a simple cell has been estimated at around 10 to the 12th bits, comparable to about 100 million pages of the Encyclopedia Britannica. And I would say, in the last 30 years, we've learned there's a lot more information in there than he ever dreamed. And by the way, Encyclopedia Britannica doesn't have many pictures. There's a massive amount of information in every cell. Folks, it is more complex than New York City, with everything in New York. Way more. Over 200,000 molecules are being built per second. You're sitting there listening to me. Why do you think you're getting tired just sitting there? Every cell in your body, and there are billions of them, have been making 200,000 molecules every second! I hope you ate well today. Because a lot of amino acids are being used up. Now, I got to tell you this, but I've been talking too long, so we're going to take just a second here. Everybody's going to stand up for me because I need your attention. You're getting tired, and I don't blame you. I'd be tired too if I was sitting there. So you're not allowed to leave, but I want you to stand up, and we're going to wiggle. Be frenetic. Do a little bit of frenetic. Thought about leading you in jumping jacks, but maybe not. Okay. Thank you very much. That'll get your blood flowing and you can make more proteins. Here's the next little book I'm going to introduce to you. The Myth of Junk DNA by Jonathan Wells. 2011 publication date. Very current. Did you know, class, that starting in about the late 1990s, we started to learn more and more about the DNA, and it started becoming obvious there were parts of the DNA that didn't appear to be being used. In fact, it became clearer and clearer in the early 2000s that only about 5% of the DNA was used to do what I just described to you. Are you listening? About 5% of the DNA was used to build protein. So it appears. And so what started to be said was, what about the rest of that DNA? People would say, only about 5% is used for building protein. What about the rest of it? Well, they would say it's 95% junk. And I can read you quotes out of this book and lots of other places from scientists who say those who believe in creation need to take a second look. Why would some God make DNA and it's 95% junk? Doesn't look like an evidence for God to me, they would say. Oh, the 5% does some interesting things. I just described it to you. But if 95% of it's junk, that sure doesn't look like a design. So in the early 2000s, this began to be talked about. You have lived through the time, ladies and gentlemen, that we have mapped the entire human genome, right? By the end of 2003, Francis Collins and his team had led the parade, and they had completely mapped the human DNA. You remember this. Everybody in this class was alive except in 2003. Folks, that was a life-changing time in our history. Now we have the whole DNA mapped. We know what's in there. And did you know we're moving toward the time when they can take your DNA and map yours? Because everybody's is unique. And if we ever get to that point, back in the early 2000s, this cost you about $1 billion to map your DNA. It's down to about 3,000 or 4,000 now. So we're reaching the time where a doctor may be able to say, okay, let's just map your DNA and let's figure out what all your genetic problems are. And using that, we can help doctor you. Wouldn't that be wonderful? I think it's nice. But I'm telling you, when that whole genome was mapped, there were a lot of surprises. There were portions of the human DNA that looked like just stretches of vast wasteland. Just repetitive junk. And we knew not anything about what was going on in there, except it sure looked like junk. There were parts of it that looked like, they call them pseudo-genes. Because it looked like they used to function as genes, but now they're not working. Give you one illustration. In your DNA, there is a pseudo-gene for vitamin C, or so they tell us. Did you know your body doesn't produce vitamin C? So you better be taking it. Did you know rabbits do produce vitamin C? And so do quite a few other lower creatures. In our DNA, there looks like there's a section in there that is a pseudo-gene for vitamin C because it's so similar to the ones of these other creatures. But it doesn't function anymore. Now, here's what Dr. Francis Collins and other evolutionists would say to you. They would say, why would God design something that doesn't work? Why would we have pseudo-genes? Good question. Lots of other things unfolded. I don't have time to tell you about it. But let me just tell you my experience. I didn't know how to answer that. And I've studied this a lot. It looked like pretty damning evidence to me for a believer in God. And that's happened to me several times in my life, ladies and gentlemen. Young people that are going through high school. In my high school days, I had teachers that told me, you kids in this class who still believe in God, better get over it. We've already discovered the structure of DNA, and it won't be long till we're producing life in a test tube. You don't need God for that stuff. Did I know how to answer that? No, I didn't. And it shook me to the roots of my being. Why do you think I do lectures like this? I needed it when I was your age, and I didn't get it. And I was shaken by all these intelligent people telling me, you don't need a God for all this. Nature does this stuff. So now this comes along and throws me for a loop again. Now I'm not losing my faith over this. But I didn't know how to answer this. May I say to you, class, it's never going to be a time where you can answer everything. There's always going to be something that is thrown up where you have to rely on what you do have confidence in, while you wait to see, how do I figure this out? This is another grand illustration. And I can tell you, it was pounded and pounded and pounded by evolutionists. 95% junk! You still believe in God? Dr. Francis Collins would tell you from studying that genome, it sure looks to me like powerful evidence for evolution. Because it looks like leftover junk from trial and error, which is the way evolution works, not design. Well, this thing is really messing up, so I'm going to quit using it. Okay, so that's my lead-in. This next piece of what I want to say should take 2 months. I'm going to take 3 minutes. But what have we learned in the last 10 years since all this started to unfold? I'll tell you what we've learned, class. We've learned that the stuff they call junk is doing all kinds of stuff we had no idea about 10 years ago. It's being unfolded to us. It's one of the most exciting times in the history of mankind. We're finding out that DNA does all kinds of things we never dreamed of. So let me just flip through and show you some of the things in this little book. And by the way, he has thousands of references back here to specific scientific articles. You can go check every bit of this out. Don't you believe me? You go look for yourself. There's evidence that suggests DNA is probably functional, even the junk. Most non-protein coding DNA is coded for some kind of RNAs. What we've learned, class, in the last 10 years is there's a whole bunch of RNAs, not just messenger and transfer and ribosomal. There's a bunch of other RNAs. And it looks like all that non-protein coding stuff is coding some kind of RNA to do something. We're just starting to learn. Many non-protein coding DNA sequences are conserved. That means from one organism to another, there's very little change in it, which speaks to function. RNAs from this DNA play significant roles in controlling whether, where, or to what extent protein coding regions are transcribed. Those other RNAs are kind of managing things inside the system. Introns. You remember what those are, class? Introns, those little white regions that I said were snipped out. You know what the evolutionists said about that? That's just junk. Why would God make something you have to copy and then you got to edit it and splice it? Why not just make it so it's ready to copy right then? Those introns are junk. You know what we learned in the last 10 years? Those introns are doing all kinds of things. They're rich in splicing factor recognition sites. They encode a majority of the small RNAs that do all kinds of things. Introns code for RNAs, people. They're not junk. RNAs from introns influence gene expression by modifying chromatin. Don't have time to tell you about that. Densely concentrated specified information in DNA. Look, people, DNA has so much information packed in it. How do you get at it? And how is it so compressed and compacted? Here's what we're learning. There are multiple messages stored in the same sequence. Now, I've got to take just a minute to tell you about this. This is too exciting. You remember that copy that RNA made? Of a messenger RNA? And then it came out here and you had to make it mature by snipping out the intron? I'll tell you what we've learned, class. That same stretch of DNA that was copied by messenger RNA can produce up to 1,000 different proteins by being snipped in different ways and reshaped and reorganized. So the same message has been so designed that one message contains the code for over 1,000 proteins. Did we know any of that 10 years ago? No, we didn't. We called it junk. And it's those introns and those RNAs that do the snipping and the arranging it all around and that guide the process. Looks like a plan to me. Spliceosomes and editosomes, which make it happen. They are proteins. There are codes within codes. I got to give you this little example. During the Revolutionary War, there was a mama who was at home with her kids. She sent a letter to her husband, who was an officer in the army. And the letter said, "Dear Joe, good things going on here at the house. Kids are fine. Pigs are having babies. Cows are doing well. We had good crops this year." You know, just that kind of stuff. Telling about home on the farm. But inside that message, if you knew how to decode it, there was a message about how many British soldiers were in the region, where their munitions were, and what was going on with their movement. If you knew how to read it. So there was a code inside the code. You know, we got that exact same thing inside the DNA now. That's what we're discovering. Now you tell me, class, the blind forces of physics acting by natural selection, are they going to produce a code inside a code? That is unbelievable. But that's what we're learning. Dual and overlapping messages all over the place. Pseudo genes I already talked to you about. What we're finding out is those pseudo genes have functions. They're not just sitting there. They do things. And we're just beginning to learn. The repetitive non-protein coding DNA, they're called long interspersed nuclear elements and short interspersed nuclear elements, lines and signs. And what we're learning, people, is they have a critical part to play in managing the whole system. You kids are good at using computers, right? Every computer to function has to have what? That runs the whole computer. What do you call it, class? An operating system. Exactly right. I'll tell you what we're learning is the rest of this DNA, it appears, is a giant operating system that's managing everything that's going on. Looks like a design to me. You think operating systems were designed by somebody? Bill Gates, maybe? Endogenous retroviruses, we're learning a lot about those too. Files within folder systems, there are words, sentences, and paragraphs that are called genes, gene folders, and superfolders, and ISO cores. Listen, we haven't begun to understand how that DNA organizes itself. Well, how does your desk look at home? You got lots of information. I bet it's a mess. DNA is not a mess. Everything's organized. So if you need to find this piece of that piece, and within that piece, you can go right to it. I will tell you, group, if we received a message from space containing just the genetic code and an explanation of how to use build proteins, everyone on Earth would say the source was intelligent. No question. So which is more reasonable, class, from what I've described to you today? I would say with Dr. Behe, everywhere we look, from the macroscopic to the microscopic, things look like they're made. Look like they're designed. And I think the simplest explanation of that is they were designed by a grand designer who had us in mind. And that's my lesson for this hour.